Let's thank Dr. Canad for having us. Thank you very much. Our final session this afternoon is Dr. Tim Sharbel. Tim is the Research Chair in Seed Biology for the Global Institute of Food Security right here in Saskatoon. He's had a significant research career working at McGill University in Montreal, time at the Max Planck Institute, and today he's going to talk to us about one of his specialties, Apomixis. Welcome.
So I'd like to thank the organizers for inviting me to give a talk here. And before I begin my talk, I'd like to thank my group. I'm in the process of moving my research group from Germany to GIFS, and these are the people that do all of the hard work and generate all of the cool data, and so I'd like to start by thanking them. So I like to talk about sex, and I thought I would start off with a 2-minute short animal porno flick. And so, I'm showing you this film for a point. So I promise you'll understand why I'm showing you this. So the context of this is that these are about 1 centimeter long flatworms that live on the Great Barrier Reef. They're hermaphrodites, but they can't self-fertilize themselves. So you have to fertilize another individual, and the way they do it is that their penises have evolved into little syringes, and these syringes can be used to inject sperm anywhere in the body of your partner. Okay, so that's how you reproduce. So let's do the film. It's just 2 minutes. Do I start it, or can you guys start it? It's a National Geographic film, so we can just watch it.
Flatworms have both male and female sex organs. But when it comes time to make little flatworms, they have to decide who plays which role. And that they fight over.
There you can see the penises coming up there.
The two flatworms rear up, exposing their midsections. Those jutting white nibs aren't weapons, at least not intentional ones. They are actually the worm's penises, a double-barreled inseminator, if they can shoot first. Flatworm sex consists of the two attempting to stab their lover with their pointed pair and inseminate each other, an act delicately referred to as penis fencing. But there's nothing delicate about it. Flatworms have been known to gouge holes in each other in battles lasting up to an hour. Why do they cross swords? Producing offspring requires a lot of energy. The winning father can go out and about in the world without any further responsibility. The losing mother absorbs the winner's sperm. It must now work longer and harder to find food and stay alive. The father floats merrily along. The mother crawls along looking for a meal, already the responsible one.
So not too far off from humans. I guess so why did I tell you that? So why I told you that is, I mean, this is an extreme example of sexual selection. And the point I'm just trying, and you can see from these pictures, is that these things actually rip each other to shreds when they're reproducing. So, and the point I'm just trying to make is that natural selection for sexual reproduction is an incredibly strong force. And when you go to look at plants, for example, so on the left-hand side, this is just, we all know what sex is in plants. It's very well defined, very highly constrained, and very difficult to find variability upon which we can act upon, right? And in apomictic plants, so I'm going to talk to you a little bit about why Apomixis is such an important technology for us to undertake. But Apomixis, so on the right-hand side of that diagram, you can see that rather than having genetic material coming from the mother and the father, the female actually makes a meiotically unreduced egg cell, which develops into a genetic clone of herself. And males actually have very little to do with reproduction. And so the other point, besides the goal of putting Apomixis into plants, what I would like to say is that Apomixis has a wealth of information in it. They're natural mutational laboratories that enable us to look at pollen formation, egg cell formation, endosperm formation, and so on and so on. So that's just my little thing. All right, but I'm standing here because, how can How can refraining from sex help humanity? So I think we've all seen this, and people have alluded to this today, right? So many of our crops depend on hybrid technology, and hybrid technology and exploiting heterosis leads to yield increase. Now, in things like, in pulses where you have cleistogamy, it's very, we haven't been able to exploit heterosis because crossing schemes are very difficult in plants that fertilize the closed flowers. So, so the reason we're studying Apomixis is because Apomixis is a tool that could lead to a fixation of heterozygosity and hopefully heterosis in crop plants. And so this is just, we know this is a typical breeding scheme. The thing is, if a farmer takes those, grows the F1 plants, right? They get the seeds from the companies, they grow the plants up, the plants are all genetically uniform, but then that farmer takes those plants and crosses them again, then sex and meiosis just mixes everything up. So you lose your traits, you have increased variability in the next generation and so on. So, what it means for the farmer is the farmer, of course, buys seeds every year. If the idea that we're working on is that if you could take that F1 population and then simply turn sex off and turn on Apomixis, then those plants would reproduce clonally from then on. And while it sounds really bad for companies, it's not real. I told you that last statement about buying seeds one time and never buying them again. That's what most Apomixis researchers have been saying this for years, right? And so I don't believe that. I'm just stating it. But nonetheless, when people talk about the introduction of Apomixis technology into crops, they tend to use the word billions. So it would change things. It's a very disruptive technology. So on the left, if we think about hybrid crops now, we have a significant investment required, time and resources to create, to recreate these F1 hybrids every generation. And the idea would be that if we could introduce Apomixis, you could make single generation hybrids, right? So you could cross plant X with some distant germplasm, let meiosis and sex mix things up for you, right? Throw the seeds out into some selective regime, and that could be temperature or light or whatever you want. You choose the best phenotype that grows out of that in that selective regime, turn off sex, and go to seed increase right away. So that and so the idea would be that we could both quicken the pace of the generation of new hybrid plants, and secondly, we could utilize a lot more of the biodiversity in the world. So I think really when I think about Apomixis, I think about diversification. So we think about single generation hybrids. We could potentially produce unlimited numbers of hybrid crops. It brings up the concept of niche breeding, and furthermore, I think farmers, local farmers who have locally adapted types of crops those farmers could actually be brought into breeding programs, because we could very rapidly breed or exploit some of these traits using Apomixis technology. So there's local and global benefits. So my research group, how much time am I doing here?
You're doing well. You have about 15 minutes.
Okay, so in my group, we basically approach Apomixis, understanding Apomixis. We look at naturally occurring apomictic plants, and our goal is to really utilize a lot of the literature in human disease research. And so what we endeavor to do is to understand these natural populations of asexual plants or apomictic plants. We very well define the phenotype, and we're really getting down to cell-specific analyses, right? So we're interested in pollen formation and egg cell formation and so on and so on. I work on a number of species. The one I'm going to talk to you about today is one I've been working on for almost 20 years. So the genus is Buchera, or Buchera. It's a crucifer. It's related to Arabidopsis thaliana. A lot of genome synteny there. It's closely related to Brassica. And the idea would be that if we can identify genes for Apomixis in Buchera, then we could apply them relatively rapidly to Brassica. So let's just talk quickly about phenotypic variability. So an apomictic plant it's not an on and off, it's not a 0-1 trait. Very often apomictic plants are facultative, so they produce a certain proportion of their seeds apomictically, which means that when you're analyzing the phenotype, you have to sample many seeds from a single plant to understand what the penetrance of the trait is. And so we do this using flow cytometry. And so this just shows you normal sexual reproduction. We have double fertilization in angiosperms. So one sperm cell is used to fertilize the egg cell. One sperm cell is used to fertilize the binucleate central cell. And you end up with an embryo that is diploid and an endosperm that is triploid. That, and furthermore, endosperm formation, you have 2 maternal genomes and 1 paternal genome. There's tons of genomic imprinting involved in that process. And typically, when you tweak any of those genes, it leads to seed abortion. So we don't learn anything from these plants. Now, interestingly, an apomictic plant, in the case of Bucera here, can produce, on the left side, it can produce certain levels of meiotically reduced pollen and meiotically unreduced pollen. And furthermore, the same plant can produce meiotically reduced egg sacs or ovules, and meiotically reduced and unreduced. By definition, there's no fertilization of the egg cell, as you can see with the big X's in the bottom, but very often we have fertilization of the central cell. And we could talk about that, but I'm not going to go into details here because of my time. The point being that when you look at the seeds of an apomictic plant, you're looking for deviations from the 2 to 3 genome ratio of the embryo and the endosperm. And furthermore, you're doing this by looking at hundreds of seeds from a single plant. So a couple of years, or 2007, one of my postdocs, Wally, we developed a high-throughput flow cytometric method. This is a paper we published in 2010, and it's simply, on the y-axis shows you penetrance, so percentage of seeds that are produced apomictically, 1 being 100%, 0 being sexual, or 0%. And every dot represents the average of 3 genotypes, 3 individuals of a genotype. And I've ranked them from low to high. And what you can see is that we have obligate sexual things at the lower left. We have obligate apomictic things at the upper right. Circles are diploids, squares are triploids. And furthermore, we have a little bit of leakiness, so facultative apomixis, so we have things that produce 5 to 10% apomictic seed at the bottom, and things that are producing 80 or 90% apomictic seed at the top. Why is this important? It's important because apomixis, it starts in a developing ovule, right? So when we're and I'm going to show you in a minute, when we're actually doing our omics analyses of these developing ovules, you have no idea when you're microdissecting a cell whether that egg cell is going to be apomictic or sexual. So very importantly, having this kind of data enables us to choose things that are 100% apomictic and compare these things to 0% apomictic. So we know we're actually comparing those two traits, right, without any mixture of anything that's not reproducing apomictically. Quick time? How much time?
Do not be alarmed, but 10 minutes.
10, perfect. No, we're right on time. Okay, so we do these analyses, right? Oh yeah, the last thing I want to say is that graph I showed you, right? So my postdoc, Wally, he analyzed 22,000 single seeds. This took months. It's unprecedented. People have not been able to do this in the past, right? So it's an incredibly informative analysis that enables us to really understand, to try to connect these thousands, if not millions, of polymorphic markers with penetrance of the trait. It's a very, very important thing to do. All right, when you do those analyses, what we discover is that 95% of the diploid apomictic Bucera that we've analyzed produce seeds with a diploid embryo and a hexaploid endosperm. Now, why is that? Well, it tells us two things, potentially tells us two things. It tells us that these things are producing meiotically unreduced egg cells and sperm cells, and for some reason, there seems to be selection for this hexaploid endosperm. Now, you recall when we talk about sexual endosperm, we have 2 maternal genomes and 1 paternal genome to make them triploid. In an apomictic plant that has a hexaploid endosperm, we have 4 maternal genomes and 2 paternal genomes. So we don't disrupt the ratio. So we think this is one of the reasons why there's a maintenance for the fertilization of the central cell. So the hypothesis is meiotically unreduced egg cells and meiotically reduced pollen cells. This is the first step in what we think is in the pathway to apomictic seed development. So we decided to have 2 projects. One was led by one of my postdocs, Jose. We're interested in the female part of the trait, and what we do is we don't do laser capture or anything. We do live microdissection. We're trying to minimize degradation of mRNAs and things. And what we can do is, on a good day without not too much coffee, you can go in and you can actually micropipette, like 10 or 20 ovules, and we can get a good transcriptomic analysis of that. So we did that this was done like in 2009 or something like that. And we go to microarrays. We're not using NGS at this time. I can talk to you about that later if anyone's interested. But the important thing is that we're comparing multiple genotypes of sexual individuals and apomictic individuals. Now, if you compare any 2 things, they're going to be genetically related to a certain extent, but you're always going to find differences between them. So by having lots of genetic backgrounds of sexuals, lots of genetic backgrounds of apomix, what you're doing is statistically looking for things that are the same in all sexuals, but different, and yet the same in all apomix. And when we did this analysis, corrections for multiple tests, and so on, we find 4 probes on our array, and lo and behold, those 4 probes nail 1 gene. So it's like technical replication, biological replication. Everything points to this gene as an important gene. My postdoc called it Apollo. I didn't. I don't take responsibility for that. But to make a long story short, if you do the sequencing, sexual plants have sexual alleles, and apomictic plants always have a sexual allele and an apomictic allele. And the apomictic allele differs by a functional polymorphism in the 5' UTR. Importantly, if you look in somatic cells, so roots, leaves, and so on, the gene Apollo is always expressed. So it's a housekeeping. It's associated with the cell cycle. And more importantly, in sexual plants, Apollo is turned off, whereas in apomictic plants, it's turned on by expression of the apomictic allele. So and we published this a few years ago. Now, we're interested in evolutionary biology and population genetics. Conservation means importance, and in red, so we've looked at apomictic bucera from all over North America. Blue is sexual, red is apomictic, and red demonstrates the conservation of this Apomixis allele across North America. So it's highly conserved, and therefore we hypothesize that it's very important. All right, that's the female story. Talk about the male story. This is done by a PhD student at the time in my group, who's now a postdoc in Germany in my group, and we were looking at anther head formation. And so Martin spent a lot of time trying to identify at which point do sexual and apomictic anthers differ with respect to meiotically reduced or meiotically unreduced egg cells. We went back to our microarrays, same kind of experiment, completely different tissues and so on. This time around, we nailed 3 probes on the array, and those 3 probes hit 1 gene. So now, is it the same gene or is it a different gene? It's a different gene. So Martin called this thing upgrade, unreduced pollen grain development, and I'm not even going to begin trying to explain this diagram to you. The take-home message, though, is that this is essentially the first Apomixis-specific gene ever identified in plants. This thing, it's a chimera between at least 3 other genes that we've been able to identify, and furthermore, the original genes to which this chimera corresponds are found in both sexual and apomictic plants. So we think that this gene is post-transcriptionally regulating other genes in the genome, and it's very difficult to explain its origin because of this chimeric structure. And finally, most importantly, as with Apollo gene, this upgrade gene is highly conserved across North America. Now, you recall at the beginning of this part of my talk, I said that most apomictic plants make a diploid embryo and a hexaploid endosperm. Therefore, we need meiotically unreduced egg cells and pollen cells. I've shown you 2 different genes which are highly correlated with the production of those 2 things. Therefore, if our hypothesis is to be supported, we would expect both these genes to be found together. And lo and behold, if you look in black across North America, these are all individual plants that are apomictic that share both of those genes. So once again, we think that this is highly suggestive that the genes we have are the genes, in fact, that are underlying this trait. So that's pretty much it. I only had half an hour, so I wanted to stop. So what I think is, clearly there's a lot more to the story we could talk about, right? So I was just being quick and showing you some of the highlights. But we really do think we have our foot in the door. And so I've shown you that there's a separate mechanism for, apomictic or apomeiotic egg cell and pollen cell formation. There apparently is a different factor for both of these things. And what we're looking at right now is to try to figure out where they came from. And what our impression is that these things have been independently derived during the evolution of bucera, kind of in a post-Pleistocene glaciation context of North America. What we are doing, though, is we have some, besides my funding from GIFS, besides my funding from Germany, I also have some venture capital funding. And what we've just received a lot of seeds from Arabidopsis and maize, and we're awaiting brassica right now with these codon-optimized constructs containing Apollo. And what we're trying to do right now is induce the trait or anything that resembles that trait in these plants. And with that, I think I'll say thank you for listening. Give you a little bit of Darwin.
Questions for Dr. Sharbel? Yes. If you shout it, maybe we can repeat it as well.
Thank you. So I have a flippant comment and then a question. So my flippant comment is, of course, Apollo should have been Aphrodite.
Good point. But it didn't match.
And secondly, if you look at the evolution of land plants, and you look back on all the genomes of the very primitive of the first of the plants to make it out of the primeval soup, how ancient is upgrade and how ancient is Aphrodite?
So upgrade only exists in apomictic bucera. It's not found in any other plant, first of all. Okay. Apollo.
So it's not found in mosses?
Upgrade is a chimera. So it was literally, sorry, this occurred in Bucharest. So sometime during the cladogenesis of Bucharest, and we're looking at this now, right? It's not, so if you go, Bucharest, and then there's Bucharest, there's things outside the phylogeny, and if you start going it back, upgrade is not there. It's only in apomictic Bucharest. So it's really recently derived.
So is it by horizontal gene transfer?
No, it's as I said, it's a chimera. So it's some sort of genesis. I can't answer that. I don't know what it is, and we're trying to determine this. Now, so Aphrodite, this gene is found, in the plant kingdom, and so it's in fact there's orthologs even in animals if you go, if you relax homology constraints and things like that. So it's a gene associated with mitosis, and it's not the gene itself which is important, it's the functional polymorphism which is important. And without going into all the details about it, we're unclear, or what we're looking at is whether that functional polymorphism both introduces novel transcription factor binding sites into the genome, and it also removes some. And so what we're presently also looking into is the titer of these transcription factors. And we're thinking that maybe Apollo acts as a sink. Either as a sink or, or something along those lines. I'm not sure though. So does that answer your question?
Sort of, but it's great stuff. Thank you.
Other questions? Yes, at the mic.
Tim, what puzzles me is that, well, I've never heard about Bucharest, but you have all this genetic information from so many locations in North America. Why is that?
Why do I have so much information?
Well,
I mean, great collaborators.
This plant is not very well known.
Oh, no, it's not. So in 1999, I joined the Max Planck Institute for Chemical Ecology in Jena in Germany, and my supervisor at the time was Tom Mitchell-Olds, who was a visionary. And when I joined his group and I told him I wanted to work on asexuality, I was in his office, he turned around and said, "Here," and gave me 2 big boxes of seed material that he'd collected all over the place. And that was the beginning. And the Bucharest community, it's a relatively small community, but we're very tight. We're all very good friends, and we exchange material like crazy. So that's where I have all of that stuff.
Chair's prerogative. Working with those next set of various crops, what increases the candidates that you might want to use for those cassettes that you're working on or other things?
I don't understand the question.
So what's the next, so you said that you want to introduce this into various crops.
Yes, yes.
What are those priority
Oh.
characteristics of those crops that will make this work?
We'll see. So it's a little bit of a fishing experiment now, right? So we're looking just for some quick and dirty information that shows that it's doing something. I mean, meanwhile, I've got these other projects going along, right? But I think the evidence is compelling enough, or it is compelling enough, really, to say, okay, let's go fishing and just try it. But I've recently hired a postdoc from China he's my genome editing person, so we're already thinking about that. I will point out there's one final little kind of secret thing. We've discovered what apparently is an interaction between Apollo and Upgrade. So I'll just leave you with that piece of information, which is very interesting. Yeah. No, I think it's an excellent question, and it's probably a different mechanism. Well, no, it's probably different genes. Let me answer that correctly. Every apomictic species that people look at, and so we're looking at about 6 different monocots and dicots in my group, and in each of those cases, so sexuality is the ancestral trait in all of these things. So sexuality, or apomixis, was derived from sexuality repeatedly during the evolution of these different plant systems. Now, apomixis tends to deal with modifications of meiosis. So clearly you only have kind of a certain set of genes to work with, right? So definitely it's conserved in that sense, right? But the mechanism to start the whole process off, I think in a neo-Darwinian fashion, so it's a mutational process, right? So it's probably a different kind of starting gun that kind of generates the process each time. And hence the reason why we want to look at so many different apomictic species, because each of those represents a different toolbox in my perspective.
Let's thank Dr. Sharbel.
Thanks.
I've got to say, this is the kind of session that reminds me why I love agricultural science. We are back here at 3:45 for the next section of crop performance and nutritional quality. The standard snacks and reception are out there, or at least the break, and then we're back here at 3:45. Thank you very much for your participation.